Hello, my name is Joshua Weber, and I am a biology education major in my third year at Concordia College.
And I am Hannah Olson, a rising sophomore at Concordia College. And this summer we were able to be part of the Wiley Lab group. And for our project, we worked with bioplastics. Specifically, we wanted to improve our understanding of chitosan alginate bioplastics and how they release and uptake different materials in a variety of solutions.
Bioplastics, by definition, are biodegradable plastics made or derived from biological materials. Our bioplastics were specifically made of chitosan and alginate. Chitosan is a derivative of chitin, which comes from the cuticles of crustaceans, insects, and fungi. Alginate comes from seaweed.
Chitosan and alginate are long polysaccharide chains, and by themselves, they don't really give rise to a strong and sturdy plastic, but by manipulating them and having chemical reactions to cross-link these chains, we can create an interconnected matrix of chitosan and alginate, and this will give rise to a plastic that is stable and strong and also made entirely of biological materials.
1.5 grams of chitosan and 1.5 grams of alginate powders were mixed together. These powders were added to 100 milliliters of RO water. The powders were mixed into the liquid until a suspension was created. Then the dye or other additive for the specific experiment was added.
And then 100 milliliters of 1% lactic acid was added to the mixture. Once the mixture had thickened, it was separated equally into 4 petri dishes. 0.1 molar calcium chloride was added on top of these petri dishes and then poured off after 1 hour. These dishes were allowed to cure and dry for 1 week.
Once the bioplastics were prepared, systems of uptake and release were created. Bioplastics were harvested into strips of uniform mass and placed into vials containing solutions created to test a specific property of the bioplastic.
For release systems, the bioplastic had color in it and the surrounding solution did not have any color. So we would measure the surrounding solution's color absorptivity to see how much was released. But for uptake systems, bioplastic was colorless and the surrounding solution was colored, and we would see how much color was uptook from the solution into the bioplastic by measuring the absorptivity of the surrounding solution.
While many of our experiments can be analyzed by simply observing a change in color concentration, we were able to accurately quantify our analysis using various pieces of equipment. For most of our food dye data, ultraviolet visible light spectroscopy was used to catalog the change in a solution's concentration by measuring the absorbance.
High-performance liquid chromatography, or HPLC, was used in the analysis of some of our solutions. We did use it for some of our food dye solutions, but mostly for our colorless solutions like our sulfa drug experiments. We were also able to use infrared spectroscopy for some of our experiments as well.
Going into this research, it was an assumption of ours that all food dyes would interact with the bioplastic in similar ways. But after a couple of initial experiments, we discovered that not all food dyes behave the same. For example, Blue 1 food dye releases and uptakes exactly as we would expect it to, but Red 40 barely releases from that bioplastic and crashes out of solution in our uptake experiments. This brought us to the idea that the structure and charge of what is being studied, whether it be food dye or sulfa drugs, influences the interaction with the bioplastic.
In addition to the structure and the charge being studied, the nature of the solvent also impacts uptake and release. By changing the salinity of or the type of salt used in the environment of the bioplastic, the release and uptake can be affected greatly. The polarity of the solution also has a great impact on the release and uptake from the bioplastic.
Finally, the nature of the molecules being studied also impacts release and uptake. All of the food dyes we used are sodium salts and therefore ionic, while substances like the sulfa drugs are very much covalent. This difference in bond type is also thought to have influenced the release and uptake.
The science of bioplastics is a fast-growing and exciting area of research. While in the Lively Lab we have tested analgesics and sulfa drugs, there are still many more factors to be explored. For example, bioplastics can be used to pick up heavy metals and other pharmaceuticals, and there are even more environmental factors such as pH and temperature to be explored. Thanks for watching, and a special thanks to ERSCA and Centennial Scholars for supporting our research.